Throughout this presentation, I will go over how to streak out a plate, gram staining, catalase tests, and spore staining. First, we will go over how to streak out a plate. We will be using a blood agar. Blood agars are enriched medias containing 5% sheep blood, but you may also streak your bacteria out on chocolate agars, McConkey's, enteric, mannitol salts, and several others. To begin, have your blood agar plate and your bacterial sample ready. Using your bacterial sample, place a dime-size amount of bacteria onto your blood agar. Then, take your inoculating loop and sterilize it in a Bacti incinerator or the flame of a Bunsen burner until it turns a bright orange in color. Once your loop is sterile, make sure it is cool by stabbing it into the agar off to the side away from any bacteria. Then, with one side of your sterile loop, make a zigzag pattern as so. Then, use the opposite side of your loop to create another pattern off of your previous one. Re-sterilize the loop and repeat the process twice more until you finish in the middle. The purpose of this zigzag pattern is to dilute the bacteria and hopefully get individual colonies. Once you've finished streaking out your plate, put the lid back on and place the media upside down in an incubator at 37 degrees Celsius. Now all that's left to do is wait 24 to 48 hours to see your results. After 24 hours, this is what your plate should look similar to. Notice the individual colonies. Now, you're going to want to look at the colony morphology and determine the hemolysis pattern. The different hemolysis patterns you may see include gamma, which is no hemolysis at all, alpha, which turns a greenish color under the colony, and beta, which appears as a complete clearing around the colonies. Next, we will go over how to do a gram stain. You will need a clean slide, 4 stains including gram stain crystal violet, gram stain iodine, gram stain decolorizer, and gram stain safranin. You will also need distilled water and a Bunsen burner. First, place a small drop of distilled water on your slide. With a sterile inoculating loop, take a colony from your bacterial plate, as so, and then mix it with the water droplet on your slide. After that, you will run it through the flame of the Bunsen burner until it dries completely. Once your slide is dry, you may begin the staining process. First, we will use gram stain crystal violet, which binds to the peptidoglycan layer. Flood the slide with the stain and let it set for 10 to 60 seconds. Then rinse the stain from the slide using tap water. The second stain we will use is gram stain iodine. This stain binds with crystal violet to help secure it within the cell wall. The second stain we will be using is gram stain iodine. This stain binds with crystal violet to help secure it within the cell wall. Flood the slide once again and let it sit for 10 to 60 seconds. Don't forget to rinse. Next, we will be using a decolorizer to remove the stain and to hydrate the cell wall to help it retain crystal violet. You do this by adding just a few drops to the slide. Once you have administered 3 drops of your decolorizer to the slide, rinse your slide again. Lastly, we will use gram stain safranin. This is used to counterstain the sample. Since the safranin is lighter than crystal violet, it doesn't disrupt the purple coloration in gram-positive cells. However, the decolorized gram-negative cells are stained red. Rinse the slide with tap water once more and then let dry completely. Here is an example of gram-positive cocci. This is an example of gram-positive bacillus or rods. And this is the example of gram-negative bacillus or rods. Next, I will go over how to do a catalase test. What you will need is a clean slide, 3% hydrogen peroxide, a pipette, bacterial sample, and an inoculating loop. First, have your clean slide ready. Using a pipette, place 1 drop of hydrogen peroxide on your slide. Then with your inoculating loop, pick up a colony from your agar and place it in the hydrogen peroxide. If it bubbles, the test is catalase positive. If no reaction occurs, the test is catalase negative. Lastly, I will go over how to do spore staining. You start off the same way you did with the gram stain. On a clean slide, place a droplet of distilled water and mix your bacteria with it, and then dry it over the flame of a Bunsen burner. Then, cut out a small piece of filter paper and place it over your dry bacterial sample on your slide. Then flood it with carbonyl fusion. Next, allow the slide to set over steam for 3 minutes. Once your 3 minutes are up and you remove the filter paper, this is what your slide may look similar to. Then apply a few drops of 1% sulfuric acid to the slide. Once you have removed some of the stain with your 1% sulfuric acid, you will flood your slide with methylene blue. Then rinse the slide and allow it to dry completely before evaluating. If your sample is spore forming, this is what you may see. This is a picture of bacillus.